droplet digital PCR for oncogenic KMT2A fusion detection in AML. This is the citation. It was published at the end of 2023. So current diagnostics and issues for AML, current MRD detection methods for AML include bone marrow morphology, flow cytometry, and DNA sequencing among many diagnostic procedures. But all of them have their various issues. Bone marrow morphology detects leukemic cells with 5% limit of detection. Flow cytometry is more sensitive but difficult to implement and lacks standardization. DNA sequencing can identify cells by mutation, but is costly and may be affected by clonal hematopoiesis. Currently, RT-qPCR can find oncogenic fusions that can cause CML by looking for their unique patterns, so this has been implemented in other diseases. And a newer test called ddPCR is even better because it's easier to do and can find really small amounts of these fusions and provide clear results without any extra steps. AML is another kind of blood cancer but has more complex oncogenic fusions to track, unlike CML. Thus, it is pretty hard to implement this test for AML, so people are looking for various new methods that we can use for diagnosing AML. Therapy-related AML or TAML is a specific type of AML that develops after treatment with chemotherapy or radiation for other cancers. Sometimes the treatment itself can trigger the formation of harmful gene combinations, initiating cancer, and specifically a group of drugs called topoisomerase II inhibitors are linked to dangerous gene combinations involving the KMT2A gene, which can lead to aggressive leukemia. Initial experiments in mice have shown that these KMT2A gene combinations can turn healthy bone marrow cells into leukemia cells. And for patients treated with high doses of topoisomerase II inhibitors, developing TML is serious and often a deadly risk. So detecting these gene combinations early could be effective for intervention. And unlike many cancer-causing gene combinations, there are over 80 different partners known to fuse with the KMT2A gene. About 80% of these harmful gene combinations involve just 5 specific genes, but due to the variety of fusion partners, traditional PCR methods struggle to detect these gene combinations effectively. So this paper introduces a new ddPCR assay designed to identify the 5 most prevalent KMT2A fusions found in TAML patients. The described ddPCR assay is cost-effective, quick, sensitive, and precise for the detection of KMT2A fusions, and this assay has the potential to enhance monitoring of minimal residual disease in AML patients with KMT2A fusions and to screen individuals at risk of developing TAML after their treatment for cancer. So I'll quickly go through the materials and methods. So for cell lines, they utilize human cells with known KMT2A fusions for assay design and validation. Various culture media specific to each cell line was employed for optimal growth. Patient samples were acquired from seropreserved human samples with TAML with informed consent under approved institutional board protocol. For RNA extraction and cDNA synthesis, extracted RNA from cell lines and patient samples. They extracted it using a commercial kit, and they quantified RNA and synthesized DNA using a standard kit as well. The ddPCR primer and probe design, they designed primers and probes to target the fusion points in the KMT2 gene and its partners, and they validated the design by sequencing cDNA amplicons and mapping them to the reference genome. For the reaction conditions, they performed the ddPCR using a specific system in supermix with precise primer and probe concentrations. They generated droplets, amplified DNA, and analyzed the results using software. For the pooled assay conditions, they created pooled ddPCR assay to simultaneously detect all 5 common KMT2A fusions. They concentrated the primer probe mix to ensure all correct final concentrations in the ddPCR reaction were correct. For the dilution series experiments, diluted cells with the KMT2A fusions, they diluted cells with KMT2A fusions into cells without fusions to simulate varying levels of disease. They extracted RNA, synthesized DNA, and adjusted the cDNA concentration for analysis. And for KMT2A fusion generation by gene editing, they used CRISPR to introduce KMT2A fusions into cells with wild-type KMT2A, and then they performed nucleofection cultured the cells and used ddPCR to detect the fusions. And for data availability, no new sequencing data was generated. All protocols were detailed in the material and method section. For the results, so they developed a dual-color ddPCR assay for KMT2A fusion detection. The assay design allows for differentiation of fusion transcripts from wild-type KMT2A transcripts. They validated the assay using cell lines with known KMT2A fusions in cells, demonstrating the detection of abnormal genes over a wide dynamic range, and they confirmed the specificity of the assay by showing no off-target detection in cell lines without KMT2A fusions. They created a pooled assay capable of detecting the 5 most common KMT2A fusions in a single reaction. The assay successfully detected KMT2A fusions in cell lines and patient samples with no false positives in control samples. They conducted experiments to determine the assay's limit of detection by serially diluting cells with KMT2A fusions, and demonstrated the assay's ability to detect rare KMT2A fusion transcripts across several orders of magnitude. And as for the gene editing, they generated the KMT2A fusions using CRISPR and confirmed their presence over time using the ddPCR assay. They also observed the proportion of KMT2A AF9 fusion transcripts that remain consistent during the culture period. So in basic terms, the results showed that the researchers created a reliable test that could find small amounts of specific abnormal genes in blood cancer cells. The assay's utility is demonstrated in both cell line models and patient samples, indicating its potential for clinical application in monitoring and early detection of TAM. So some advantages of this new method that they've tested, this new method was evaluated against older techniques, specifically bulk qPCR, and was found to be more sensitive and able to detect very low levels of genetic changes. This assay is highly specific, meaning it can accurately determine if the genetic changes are present or not without mistakenly identifying them in samples where they do not exist. It is user-friendly, so it provides precise measurement of the genetic material, allowing for simultaneous analysis of multiple targets. It can easily be adapted or expanded, and produces consistent results. Sorry. Unlike previous methods, this assay does not need a standard curve for calibration, as it can directly count the number of genetic changes. So some limitations. This test can identify a subset of 80 possible genetic changes involving the KMT2A gene, but it doesn't cover all of them. It is just designed to find the 5 most common genetic changes, which is present in about 80% of the cases. The KMT2A gene can pair up with over 80 different partners, which has made it hard to develop tests that can find all of these different combinations, but this test definitely does cover most of them. The test uses RNA, which is less stable than DNA, as it can break down more easily, which can impact the test performance. For the test to work, the genes involved in genetic changes must be active and making RNA. If they're not active, the test might not find them. If the test design doesn't perfectly match the genetic change, it might not count all of the fusions, which could lead to underestimating how much of the disease is present. The benefits of this assay are comparable with the advances for CML, where the ddPCR was used. The platform may also be used to detect early premalignant KMT2A fusions in individuals at risk of developing TAML, which is otherwise very challenging to treat. Planned studies for the future aim to directly compare this assay with multi-parameter flow cytometry and morphological analysis for assessing residual disease in AML treatment. This diagnostic test has the potential to be integrated into clinical trials that utilize this biomarker to guide treatment decisions. Yeah, that's all. Thank you.